Hello and welcome to our video. Today we are going to talk about Edwards syndrome. Edwards syndrome is a genetically transmitted disease. But to understand what that means, we need to understand meiosis. First things first, meiosis is not mitosis. Mitosis is the process that almost all cells go through to duplicate. But meiosis is the process that sex cells go through to duplicate. Now, meiosis has two parts, meiosis 1 and meiosis 2. And then each of those two parts have steps, or phases. Interphase, prophase 1, prometaphase 1, metaphase 1, anaphase 1, telophase 1, interkinesis, prophase 2, prometaphase 2, metaphase 2, anaphase 2, and telophase 2. Wow, that's a lot of phases. Let's start with interphase. Interphase is what happens most of the time. It isn't actually part of meiosis, but it is important to understand interphase. What happens is all the DNA is mixed up and unorganized, and then the DNA duplicates and sister chromatids form. The spindle fibers are made in the centrioles. During interphase, the cell goes from this to this. This is prophase 1. During prophase 1, the DNA condenses, the centromeres start going to opposite sides, the nuclear membrane starts dissolving, but most importantly, the homologous chromosomes form. Prometaphase 1 is basically an extension of prophase. The nuclear membrane completely disappears, the centrioles keep moving towards opposite poles, the spindle fibers attach to the centromeres, so the chromosomes start lining up. Metaphase 1. During metaphase 1, all that happens in prophase and prometaphase 1 is resolved. All the chromosomes are perfectly lined up, and the centrioles are on perfectly opposite poles. Anaphase 1 is maybe one of the most important moments in all of meiosis 1, because it is when the sister chromatids are pulled apart and become separate chromosomes, and the centrioles pull away from each other to facilitate the pulling apart of the sister chromatids. And finally, telophase 1. Telophase 1 is when nuclear membrane forms around the new chromosomes, and the centrioles disassemble, meaning that it separates all of its spindle fibers from whatever they were attached to. Next is interkinesis, which is a moment in which nothing happens, and the new cells simply are cells. The next thing that happens is meiosis 2. Meiosis 2 and meiosis 1 are actually the same thing, except that meiosis 2 happens in the two new daughter cells. At the end of meiosis 2, you end up with 4 new daughter cells, or gametes. When gametes meet each other, they create a zygote, or a fertilized egg. So, as you can see, there are a ton of steps, and it leaves a lot of room for mistakes. And sometimes mistakes happen. Enter genetic disease, and more specifically, non-disjunction. Non-disjunction is when 3 chromatids move to one centriole, and only one ends up with the other centriole, which ends up with dysfunctional cells after telophase. This can happen in either meiosis 1 or meiosis 2. If it happens during meiosis 1, none of the gametes will end up right, but if it happens during meiosis 2, only half of the gametes will be messed up. Here's why. Look at the duplication cycle. If it happens during meiosis 1, 6 chromatids will move into one of the new daughter cells, and only 2 will be in the other. So that when they duplicate again, there will be 3 chromatids in 2 of the gametes, and only 1 chromatid in the remaining gametes. However, if it happens in meiosis 2, only one of the gametes will have 3 chromatids, and only one of the gametes will have a single chromatid. The other gametes are normal. Now, when non-disjunction happens, a lot of things can go wrong. In this case, we are looking at Edwards syndrome. which is otherwise known as trisomy 18, because the 18th gamete is trisomic, which means it has 3 chromatids. And if a zygote has a trisomic 18th chromosome, that means that every 18th chromatid in the kid's body will have of 30, although most of the time, the affected children will die before birth. If the child is lucky and survives birth, then the average time that they stay on Earth is about 14.5 days. If the patient is extremely lucky and survives the first year, then there are few rare cases of people with Edwards syndrome surviving until teenage years. Unfortunately, these patients will suffer from severe mental and physical difficulties. Now, let's talk about the treatments. Sadly, there are no treatments for this disorder. The only thing that can be done is prolong survival and manage complications. For example, treatments may include nutritional support through nasogastric tube or directly into the stomach through gastrostomy. When limb abnormalities affect movement, physiotherapy and occupational therapy can help. Thanks for watching!